Nag-present ka na, Patrick? Nag-present mo na ba?
Yes po.
Okay, did that say other gram-negative organisms? Wala na ako mailagay na title kasi. Nilagay ko na lang other gram-negative organisms. Kasi magkakasama yan. Pseudomonas, Vibrio, yung mga ganon. Helicobacter, Campylobacter. They have a special place in my heart. Charet. Wala namang special place in my heart. Ano lang yan. Para mas ma-compartmentalize lang yung amount ng information na kailangan na makuha ninyo, class. Ganun lang yon, okay? So let's start with slide number 1. We'll talk about Pseudomonas species. Pseudomonas species are also known as your water bug bacilli. Okay? They thrive well in watery locations. Kaya nga, minsan nakikita natin siya sa luga, di ba? Makikita natin siya sa mga fluids natin sa katawan, di ba? And of course, the most popular one, Pseudomonas aeruginosa, yung nakukuha natin sa what? Sa jacuzzi. Di ba? Yung mga mahilig mag-jacuzzi. Okay? Ay, hindi ko pala napalitan yung yung geme. Ayan. Okay na, naka-live na tayo na nakikita ko. Hindi na starting soon, no? Okay? So, next slide. What is the oxygen preference? When I say oxygen preference, again, I go back to whether it's facultative, obligate, or microaerophilic, capnophilic. Okay? So, yun yung mga pag sinabi, pinag-uusapan natin. Paulit-ulit kasi laging may nakakalimot ng basics. Okay? So, the oxygen preference of Pseudomonas aeruginosa is that it is an obligate aerobe. Okay? Obligate aerobe. Kailangan niya ng hangin. I can't breathe without air. Di ba? Lagi yung sinasabi sa mga review classes ko dati. Pseudomonas can't breathe without air. Di ba? Mapapa Jason Derulo tayo dito. Buti na lang hindi ako kumanta. Wala ako sa mood. Okay? Characteristic odor produced by, I think that's the next slide. The characteristic odor by the type species Pseudomonas aeruginosa is, of course, eru siya, hindi eri ha. Palitan ko nga yan. Eru. Palitan natin nga yung Send na lang ako ng edited version, Patrick. But aeruginosa. Aeruginosa. Okay, Pseudomonas aeruginosa is, it has a, what do you call this? It has a grapefruit smell, corn taco-like odor. It has a sweet smell. It has that sweet, wonderful smell. Actually, sinabihan ako ng coworker ko dati, girl, I don't know what you're smelling, but that ain't no grape. Sabi sa akin, kasama ko. Naloka ako sa kanya. Hindi daw niya naaamoy yung grapes. Because tayo lang mga medika nakaka-appreciate na. Okay? So, yeah, it has that sweet, grapefruit odor, and taco, or corn taco-like odor. Okay? Now, at what temperature does Pseudomonas aeruginosa grow? I think that's the next slide. Okay? So, it grows well at 37 degrees Celsius. And it can grow at 32 degrees Celsius. That's how we usually differentiate Pseudomonas aeruginosa from aeruginosa na naman. Aeruginosa. Kapalitan ko yan ha. Aeruginosa. Okay. That's how we differentiate Pseudomonas aeruginosa from the other members. Okay? So you could get other Pseudomonas species, but what we're interested in is Pseudomonas aeruginosa, non-pathogenic species of Pseudomonas. Okay, so when we want to differentiate it, usually this would be if you're not using Malditov, we used to do this for Vitek. Like for example, we have a not blood culture, like we have a urine culture that we want to differentiate, that the Vitek machine detected as Pseudomonas species. There's an additional information there. Please do additional testing. So additional testing is incubated at 42 degrees Celsius. That's why we have an extra incubator there that is set at 42 degrees Celsius when I was working in Saudi Arabia. But because now we're doing molecular testing, we rarely do this anymore. Okay? Very rarely. I think sa sa almost a year kung nagtatrabaho dito, hindi pa namin ginawa yung 42 degrees. But we always do this when we were using biochemical based testing. Molecular based na kasi si Mal dito. Aeruginosa na naman. Nagigigil ako dun sa aeruginosa na yan. Okay? Alright. How would Pseudomonas aeruginosa be differentiated from other members of Enterobacteriaceae or Enterobacterialis? How? Who here knows the answer? Or let's go and do a what do you call this? A survey. Tingnan nga natin kung anong test. What test are we using? Patrick, huling-huli ka na. Nandun na ako sa dulong-dulo, Patrick. Usog ka naman ng konti, Patrick. Nasa grapefruit odor ka pa. Baba, kanina pa ako next slide, Patrick. Bakit nasa taco ka?
Wait lang po, sir. Baka ano lang yun.
Bakit nasa taco ka, Patrick?
Andun na po ako sa 42 eh. Wait lang po.
Oo, ako din dito sa may 42 na ako. Alright, so how would we differentiate? Whilst Patrick is looking for himself, hindi yan
Okay na po ba ito?
Okay. Yeah. That's the one.
Okay.
What tests are we going to use? There's two tests that we use, class. What test do we use? Sige nga. Sige. Bigay lang kayo kahit isa, class. Ano yung sabi natin? Ayun. No, nagtaas ako agad si Moyo. Sige na, Moyo. Pagbigyan na natin si Moyo. Alright, Moyo.
Ano po, sir? Oxidase test po. Oxidase positive po yung...
Sino? Enterobacterialis or Pseudomonas? Yay. Sino doon?
ano po? Pseudomonas po.
Okay, very good. Okay, what else? Moyo na naman. Ayoko na kay Moyo. Baka sabihin meron akong ano.
Pindot po, sir. Sorry po.
Si iba naman. Hindi pwede si Italia. I love the name Italia Valenzuela. Si Italia ba, may i-contribute aside from oxidase? Meron ba bang ano? Wala na. Okay, pakita na natin yung next answer natin. Oxidase, Pseudomonas aeruginosa is oxidase positive. Okay? And it is K over K. Ano yung ibig sabihin ng K over K? It is non-fermentative, meaning alkaline over alkaline. Okay, class? It is alkaline over alkaline. So please don't forget that one. That is Pseudomonas aeruginosa. Okay? Okay, class? Now, let's move on to the pigments produced by Pseudomonas aeruginosa. Ito, alam na mga students ko to. Hindi pwede. Mahilig mag-color ang mga students ko eh. Hindi ko alam sa inyo, sa batch ninyo. Previous students ko pala, I mean. Sige nga. Who here knows one of the 4 pigments elaborated by Pseudomonas aeruginosa? Mali na naman yung spelling. Nagigigil talaga ako dyan sa Pseudomonas aeruginosa na yan, ha? Aero yan. Papalitan ko yan ng papalitan. Aero. Okay. Sige, anybody knows? Kasi nag-warm up pa lang ako. Mamaya yan, bibilisan ko na. Anybody knows? Raise your hand if you know one of the few. Okay, Lil Zo. It's nice to meet you again. Okay, what's the name? I'm asking for the name. Pigments produced. The name. We're asking for the name. Marion.
Ano, sir? Pyoverdine po.
Okay, pyoverdine. Ano pa? May alam ka pa?
Pyomelanin po.
Pyomelanin.
Yun lang po, sir.
Okay, you forgot payo. What? Payo azul. Charot. Walang payo azul. Okay. Pyoverdine, pyomelanin, which is brown. Okay. And then, pyorubin and pyocyanin. Walang payo azul, ha? Char lang yan. Pyocyanin. Anong ginamit nating mga words dito? Pyo meaning pass. Diba? Pass. Cyanin, blue, verdin, green, verde. Melanin, brown, black. And then rubin is red. Okay? Now, blue pigments are produced by Pseudomonas aeruginosa. Mali na naman yung spelling. Ang gigigil talaga ako sa mali na spelling na yan. Okay? I think lahat yata yan, ganyan ang ginawa. Anyways, The blue pigment produced by Pseudomonas aeruginosa is soluble to what? Water and chloroform. Actually, the chloroform part is just an experiment. But what we want to understand, what we want is the water part. Why? Kasi class, um, kapag ka medyo humid yung pinaglagyan ninyo ng sample or yung ng plates ninyo class, medyo mamasa-masa, hindi siya nagpo-produce ng color. Nung nandiyan ako sa Pilipinas, medyo hindi controlled kasi 'di ba yung humidity ng mga incubators, lalo na pag Chipanga tayo. Hindi naman sa sinasabi kong Chipanga yung hospital sa Pilipinas, but we can still produce quality results. But the ideal conditions for these pigment producers, hindi namin na-utilize kasi nga hindi ideal yung conditions. Hindi sa sinasabi kong tsaka, huwag magpagamot dyan sa Pilipinas. Wala akong sinabing ganun ha. Hindi lang namin mapalabas yung pigments ng ibang colonies actually. Yung chloroform na yan, experiment lang yan. Dinagdag lang namin yan. Just para maloka kayo. Ganun. Okay? So Pseudomonas aeruginosa, Pseudomonas is a motile organism. Its motility is due to what type of flagellar structure. So what type of flagellar structure? Monotrichous. I was about to say peritrichous, but then I realized it's monotrichous. Okay? Next slide. Culture on blood agar shows what type of hemolytic pattern for Pseudomonas aeruginosa. So on blood agar plates, it will show beta hemolytic pattern. Okay? Beta hemolytic. Sino pa nga ba ang beta hemolytic? Gram-positive cocci, catalase positive. Sagot nga, class. Type naman tayo dyan para galaw-galaw, class. O, ayan. Sa baba ni Dayan. Ang hindi makasagot, akala ninyo hindi kayo naniniwala, bahala kayo. Ang hindi makasagot, nursing. So, ang hindi sumagot. Ano pang organism? Beta hemolytic. Another beta hemolytic organism that is catalase positive. Oh, beta hemolytic, gram-positive cocci, catalase positive. Pinibigay ko na sa inyo lahat, class. Yan ang mga dapat ninyong tandaan, no? Sinachar ko lang kayo, bahala. Ang hindi makasagot, bahala. Walang sumasagot, class. Naku. Class. Ano na class? Sheila. Italia. Sino to si Annabelle? Hindi ko mabasa yung pangalan ni Annabelle. Si Comendor. Si Daniela. Ano na? Gram-positive cocci, catalase positive. Oh. Sinong organism? Hoy, nakalimutan nyo na si Staph aureus. Catalase positive. Kayo talaga, class. Nakalimutan nyo na si Staph aureus, ha? Sasakali ko kayo. Bahala kayo, mag-nursing kayo. Pagka walang sumagot. Walang sumagot. Okay. Next, next topic tayo. Pag hindi sumagot, mag-nursing. Okay? Pathologic. Hindi, mag-BBA. Ganun na lang. Oh, bahala kayo dyan. Mag-BBA. Business ad. Okay? Pathologic determinants of Pseudomonas aeruginosa type. There are 4 pathologic determinants. Okay? And one of them is the enzymes, of course. Pili is of course needed. In this case, adherence pili, exotoxins, and then the next one is hemolysins. Bakit hemolysins? Kanina nga, di ba sinabi natin beta hemolytic siya. So there is beta hemolysis there. Natakot ka, Patrick, sa nursing. O sa BA. Ano mas gusto? Patrick, natakot ka sa nursing.
Sir, may Sophie card na nga. Dadagdag ka.
Magna-nursing. Lilzo, hindi ka sumagot. Lilzo. Ba't hindi ka sumagot, Lilzo? Gusto mo mag-nursing, Lilzo? Ayaw. Tumatako si Lilzo. Hoy, walang isahod ng mga yan. Pero char lang. Hindi ko kaya yung trabaho niya. Nakakapagod trabaho nila. Alright? Hindi ko sinasabing, mas magaling tayo sa nurses. Sinasabi ko magbabago magbabago ng karera. Wala akong nilalait. Kayo talaga, class, ha? Huwag kayong nag-a-assume. Okay? So enzymes elaborated by Pseudomonas aeruginosa, there is two. There's elastasis and proteases. Actually, in the exams, I never paid attention to this part of the discussion for Pseudomonas because I haven't encountered it in my experience as somebody who makes the exams. Okay, and yan na, may idea na kayo. So elastasis and proteases, just for your information class. Okay, next. Wounds and burns infected by Pseudomonas aeruginosa is characterized by what color? Green. Okay? Blue-green color. And that is true. Even for patients who have diabetic who have diabetic feet, yung pinutol yung paatas, dinala sa histopatas, tatahiin ng mga intern. Okay? Bluish and green ang color nila. Bluish to greenish ang color ng mga samples na gano'n na may infection ng Pseudomonas. And it's, I've seen it several times. Even the pus, there's a smell to it. Sabi nga ng pathologist namin, amoy Emporio Armani. Ganon yung sinasabi ng pathologist namin na Becky Bambam din. Okay? Anyways, let's move on to the next one. Give the other names of infections associated with Pseudomonas aeruginosa, mild otitis and skin lesions. So mild otitis is known as swimmer's ear, skin lesions is ectyma gangrenosus. Bakit skin lesions ectyma? Ectyma is actually yung characteristic na sakit ng mga isda. Ganun yung itsura niya. Pero what's the difference in humans is that parang naging scaly siya, pero gangrenous. Okay? Swimmer's ear, on the other hand, is just a mild otitis media. Okay? And again, the color of the pus that comes from the luga ng pasyente is kulay green. Okay? Next, we move on to another organism associated with Pseudomonas is with the Pseudomonas family that is known as Pseudomonas malae. Actually, now it's termed as Burkholderia malae na siya. Okay? Correct me if I'm wrong, but right now it is Burkholderia malae na. Okay? Other terms associated with that one is glanders bacilli or Burkholderia malae. Okay? So, yan. Updated pa rin tayo. Okay, class? Updated pa rin yung lecture ko. Glanders disease is an animal transmitted disease to humans. What animal usually is infected with this disease? So, glanders disease comes from horses and it has something to do with the pulmonary, with the respiratory system of the animals. So, for example, animals are infected usually with the inhalation of the particles. Okay, so glanders disease is an animal disease that is transmitted to humans. And again, this is mostly for people who work with animals. Okay, why do we need to know? Because we are Mentex. And this again, even though it's a disease of horses, it can still be transmitted to humans. Okay? So be mindful of that one. Another name for glanders disease is, again, it's in our next slide. Another name for glanders disease is known as farcy. Now, farcy, on the other hand, is a different form of glanders disease. Unlike in horses, where respiratory involvement occurs, for humans, it's more or less cutaneous. So usually, it's in the form of a skin lesion. So for example, nalawayan yung sugat mo ng kabayo. Okay, nakagat ka ng kabayo. It happens, class, ha. I know, nakakatawa, class. Medyo funny. Pero class, it happens. Okay? Okay, bahala kayo. Ang hindi makakasagot sa susunod kong tanong, magkaka-farcy. Magpupunta ng Baguio at makakagat ng kabayo. Ganun na mangyayari. Charit lang. Okay? So, let's move on to, after farcy, that's the next slide. What is the appearance of Pseudomonas malae in blood agar plates? They appear as brown colonies. Okay? So, most of the time, you would see this in wound swabs. Okay? So it's usually seen on wound swabs, and it's the characteristic brown color. It has that appearance of Pseudomonas aeruginosa colony that has biomelanin. But what's different is that it's very much circular and dome shape. Okay. Now let's move on to animal test subjects used to identify Pseudomonas malae is guinea pigs, and we inject guinea pigs intraperitoneally. What is expected for Pseudomonas malae is that the organism will have Strauss phenomenon. Okay, Strauss phenomenon. Strauss. Strauss phenomenon is swelling of the scrotum of the guinea pigs 4 days after inoculation. 1 to 2 days after inoculation, there is testicular necrosis. Okay? So after inoculation. Okay? So don't forget also Pseudomonas malae or Bacteriology malae is inoculate. We do animal inoculation techniques on this one, and we inject the pigs intraperitoneally. So imagine from the peritoneum, because of the Strauss phenomenon, the scrotum of the guinea pigs will be affected. Okay? Magsiswell siya after 4 days. Okay? That is the expected result. After 1 to 2 weeks, maluloka yung balls niya ng guinea pigs. Okay? Guinea pigs. Alright, causative agent of malloidosis is the next one. Are we there? Are we discussing malloidosis? Yeah, malloidosis. So, another name for malloidosis, the causative agent for malloidosis is Vietnamese time bomb disease. This is known as, the organism is also known as Pseudomonas pseudomalae. Dalawang Pseudomonas pseudomalae. Okay? So, Pseudomonas pseudomalae, or otherwise known, is the causative agent of malloidosis, also known as Vietnamese time bomb disease. Okay? We also have another organism that you need to remember, Pseudomonas maltophilia. Pseudomonas maltophilia is also known now as Burkholderia maltophilia, or Strenotrophomonas rather. Another name for that one is Xanthomonas maltophilia. Why did I include these other names? Is because sometimes our board examiners there in the Philippines, they don't change their names, but the contemporary nomenclature for this particular organism is Stenotrophomonas maltophilia. Okay? Please highlight that one. Let's go with, I'll change this one. Contemporary, the latest. Let's just go with the latest. As of 2024. Okay? Pseudomonas malae. Stenotrophomonas maltophilia. Okay? Now, the characteristic odor of Stenotrophomonas maltophilia is a characteristic what? Mousy odor. Or sorry, ammoniacal odor. Amoy ihi. Mousy tuloy. Iba pala yung mousy. Okay? Alright? Ammoniacal odor. Nagkamali ako. Okay? So, ammoniacal odor. We also have to talk about alkalinogenous. Okay? Is oftentimes confused with Pseudomonas, and we can use them. We can use a test to differentiate the two. That test is gelatin liquefaction. So, tandaan class, who's the other organism that? Which other organism requires gelatin liquefaction? Sige nga class, baka nakalimot na tayo class. Who's the other organism that requires gelatin liquefaction? Please type it on our chat box. Kung naaalala niyo pa class. Hoy, hindi na nagre-respond ang mga students. Paano kaya pag tinawag natin si Sir Marco? Matatakot kaya sila? Walang nagtatype. Si Alessandra lang ang nakikita ko nagtatype. Oh, si Alessandra lang. Or baka nagtatype yan ang papanoorin sa YouTube. Tulfo siguro pinapanood niyan. Bakit apat lang ang estudyante ko? Pati si Patrick. Si Patrick lang. Nasaan na si Lilzo? Si Reisha. Ayan. O, diba? O. Eto na, tatawagan ko na sana si Sir Marco. Diyos ko, buti na lang hindi ako tumawag. Baka bumula na naman yung bunganga nun. Okay? Okay. Alright. So, a lot of people answered si Reisha Marcedes. Bakit kailangan pa ulit-ulit, sir? Kasi class, memory work ang karamihan sa microbiology. Kaya ayoko kayo nahihirapan class pagka nandun na kayo. So, kung pag-uunin nyo yung sa exam, ay, mas madali pa ito sa binigay ni Sir Manuel na exam. Ano ba yan? Gusto ko ganun ang feeling ninyo kapag nandun kayo sa board exam ninyo. Okay? So, Pseudomonas aeruginosa will test positive with gelatin liquefaction. Alkalinogenous fecalis would test negative. Okay? Alkalinogenous fecalis. Okay? Now, there is a characteristic motility pattern for alkalinogenous fecalis. That's our next slide. It is characterized by sluggish motility. Okay? sluggish motility, parang tamad lang siya. Parang umiikot-ikot lang siya sa sarili niya. Ganon. Paano ko ba i-describe ang sluggish motility? Patrick, ganun-ganun, giling-giling ka nga, Patrick. Giling-giling ka lang, Patrick. Ikaw, hindi yung kapdaliri mo. Gumiling-giling ka lang, Patrick. Kaya nga, giling-giling, igiling-giling ka lang. Ganon. Hoy, baka may plus to. Ano ka ba? Kala nyo ginaganyan-ganyan ko yan si Patrick. May plus yan si Patrick. Bahala kayo dyan. Ay, naku, Patrick. Ang saya-saya mong co-host, Patrick, ha? Igiling-giling. Parang ganon. Okay? Alright, so that's the end of our discussion when it comes to Pseudomonas Now, I know there's a lot of confusion when it comes to the name, but don't forget, most of the organisms that are named today, Burkholderia na sila. Okay? Yung mga pinalta natin names. Alkalinogenous fecalis and Stenotrophomonas maltophilia. Yun lang yung mga contemporary names. Okay? Now, let's go to the next slide, which has something to do with the family of Vibrionaceae. Okay? Vibrionaceae. Now, the oxygen preference of the family of Vibrio is that it is a facultative anaerobe. Okay? It's a facultative anaerobe. Kaya nga nakakasurvive sila sa mga sugat-sugat. Okay? Now, all species of Vibrio are halophilic except for two organisms. That's the next slide, I think. Okay? So all species of Vibrio are halophilic except for two, Vibrio cholerae and Vibrio mimicus. Okay? What do you mean by halophilic? Sige nga, tingnan na natin sinong nakakaalala ng mga ating basics. Bacterial physiology, salt loving. Ayan, nagsagutan ng lahat ng tao, di ba? Dalawa lang ang students ko. Pag hindi sumagot at hindi ginaya na lang. Ganun na lang tayo kukuha ng ninyo. Salt loving, o di ba galit na galit si sino yung madaming si Aiza, galit na galit. Salt loving, ganun, galit na galit sa akin eh. Salt love lang daw. Ano yun? Salt love McVeigh. McVeigh ba to? McVeigh, gusto ko makita ko kung anong itsura mo kasi curious lang ako. Feeling ko ano to kano. McVeigh. Ano bang itsura? Magpakita ka nga, galawin mo nga ang baso, McVeigh. Pangkano yung pangalan niya o pang-afam. Diba? Parang Aaron Kirby din. Parang gano'n. Pang-afam din yung pangalan Diba? Sir, sumasayaw yan. Gumigiling-giling din yan. Igiling-giling. Okay. Huwag ka na nga, ano ba, Ronchbel? Ano bang itsura mo din, Ronchbel? Hindi ko pa din nakita yung itsura. At saka, bakit hindi sila nagpapakita? afam nga ang itsura, Mukhang opa to. Taas mo naman yung baba mo lang yung noo mo lang nakikita ko. Opa, class. Opa. Opa yan. Oh my gosh. Popompyangin ko. Opa, popompyangin ko. Ganon. Diba? Layo po. Okay. Char lang. Ini-etchos ko lang kayo para magising kayo. Diyos ko, yung mga itsura ninyo parang tutulog na. Tignan si Alessandra, Nag-ano lang yan, nag-Candy Crush lang yan si Alessandra. Ini-etchos lang ako yun Ay, kumakain. Ayan, okay ang kumakain. Okay. Hindi ako galit dyan sa ganyan. Pero yung nag-Netflix na ko, Diyos ko. Class yung nag-Netflix sa harap. Huwag nyo i-expose sa akin, nag-Netflix kayo. Okay, so salt-loving is the correct answer. So we have two organisms, Vibrio cholerae and Vibrio mimicus. Now, there are two organisms that are closely, Vibrio species are closely related with what bacterial genera? Pseudomonas and Aeromonas. Okay? Pseudomonas and Aeromonas. Okay? Pseudomonas and Aeromonas. Now, the only way to differentiate the two is using the reagent, sodium desoxycholate, which is basically, the name for the test is string test. Okay? What we do with string test is that we add it on we add it to the colonies, and then you pull it out, or use the inoculating. You dip your inoculating loop basically in 0.5% sodium desoxycholate, and you lift up the colonies, and you'll see string formation. And if that organism forms a string, that is basically Vibrio. Okay. Okay. Now, Because we're talking about differentiating Vibrio, we're talking about general characteristics. We want to differentiate Vibrio from the family Enterobacterialis or Enterobacteriaceae. What test is used to differentiate it? It's oxidase test. Okay, oxidase test. Why are we using oxidase test? Because we want to know which one do you think will test positive for oxidase test? Of course, it's going to be Vibrio. Okay? Why? Because Enterobacteriaceae are what? They are fermenters. They are not oxidizers. That's basically my shortcut. They are not oxidizers, they are just fermenters. Or these organisms are just fermenters. They are not oxidizers. Okay? So don't forget that. Enterobacterialis, oxidase negative, Vibrio species will test positive. Now, we're talking about general characteristics of Vibrio. Why not talk about the flagella or the general structures of Vibrio? Vibrio species have a monotrichous flagella and the motility pattern for this one is darting. Sige, hindi na kita pagigilingin Hindi na kita pagigilingin, Patrick. Ano naman itsura ng darting motility? Daliri, Patrick, daliri. Darting motility. Oh, diba? May sound effects. Di ba? Okay. Nakakatuwang co-host to si Patrick. Parang gusto kong gawing permanent job ni Patrick to. Patrick, magkano ang bayad natin sa'yo, Patrick? Magkano rate natin? Magkano? Libre lang pag ikaw. Okay, sige. Or ano kaya, Patrick? Diamond membership. Charing lang kayo.
Parang hawa nyo, sir.
Patrick, last na natin to. Ayaw na kitang makita, Patrick. Hindi sayang kitang makita personally. Patrick, last na natin to.
Na 30 years old yun kasi.
Oo, Patrick. Diyos ko, makikita pa tayo dito, Patrick, di ba? Ako pa nga magpo-process, di ba, ng papers mo. Assist pa kita. Di ba? Lahat ng mga CMMC ko, lahat ng mga naging students ko sa CMMC, pagka may kailangan kayo sa akin when it comes to going abroad, tanongin nyo lang ako, class, okay? Anyways, let's move on. Okay. Now, culture media used for the differentiation, for isolation of Vibrio species, we have two, okay? So don't forget your TCBS, that's what we use all the time, and also a broth medium, which is alkaline peptone water or APW, okay? So the differentiating medium or the selective differential media is the TCBS or thiosulfate biosol sucrose, and the enriched media is alkaline peptone water. What does enriched media mean? So alkaline peptone water, because it is a halophilic organism and it requires a lot of protein, this is the best media for stool samples that have Vibrio. It will kill any other organism except for that one. It will propagate the growth of Vibrio. Okay? Now, before next slide, okay? So we have choleragen. So cholera antigen toxin or choleragen toxin. There are serotypes for this one. We have 01 or O1, O139. And the action of these toxins is that choleragen acts on water and electrolyte secretion. This produces the characteristic rice watery stool. So please don't forget rice watery stool. I want you guys to highlight that one because in the board exams and even in the ACPI, you will see this terminology appear over and over again. Okay? Okay, class? Please don't forget about that one. Okay, now, there are serogroups of Vibrio. I think that's the next slide. Is that correct? Are we on the next slide? Sorry, let me double check again because, yeah. Okay, so, Patrick is still following. Okay. There are serogroups for Vibrio cholerae and we have 3. Huwag niyo kakalimutan yung iho. Ano yung iho? Inaba, Hikujima, Ogawa. They are basically names of Japanese scientists. The Inaba serogroup is found in the Philippines. The Hikujima serogroup is found in Japan, and the Ogawa serogroup is found in India. So Iho, wag kakalimutan na Inaba, Hikujima, and Ogawa. Philippines, Japan, and India. Diba? Paano basahin ni Steve Harvey yan? Philippines, diba? Japan, o diba? India, ganon. Wala yung Vietnam. Diba ang arte ng Vietnam? Vietnam. Walang ganon. Hindi, walang Vietnam, no? Nakakainis. Anyways, let's go on. Puro na naman kabadingan yung pinag-uusapan natin. There are two biotypes for Vibrio L. cholerae. Vibrio cholerae. L. cholera tuloy. Vibrio cholerae. We have the L-tor and the classic biotypes. Okay, so the classical biotypes, those are usually the ones that are not contemporary. We're using L-tor. I think what many microbiologists, no, before the advent of molecular testing, the biotypes that they used were the L-tor biotypes. And the best way to differentiate these types is using red cell hemolysis, polymyxin, Volk's proscour, and agglutination with chicken RBCs. Okay? So that's basically how you would differentiate the classical and the L-tor biotypes. Now we're going to move on with Vibrio cholerae. The name for Vibrio cholerae, the other name is the Calma bacillus. And its appearance on the gram stain is that it appears like fish on a stream. Fish in a stream. Okay? So anong fish in a stream, sir? Parang isda lang siya. Parang fish lang talaga. As in, literally parang isda. Okay? And of course, you have to indicate the result for biotypes for Vibrio cholerae. There are two biotypes, remember? So the classical and the L-tor. So the classical ones will test negative for most of the tests, or for all of the tests. And again, the latest one, of course, the latest biotype that has been discovered, or the L-tor biotype, will test positive for all. Okay? And it is resistant. Kaya talagang napaka-important pa ma-identify ang difference between classical and L-tor. Okay? Kita nyo, all of the other tests that I mentioned before is negative for classical, and the L-tor ones, the L-tor one, that is the dangerous one. Okay? Now, sucrose fermenters produce what color in the colonies of colonies in TCBS? Sucrose fermenters are yellow colonies, and non-sucrose fermenters will produce a green colony. Okay? Okay. Now, the next slide is not really a question, but more so a differentiation of what species would you expect to be sucrose fermenters and sucrose non-fermenters. Okay, so we have 5 species that we want to be familiar with, or to familiarize ourselves with. Sucrose fermenters, Vibrio cholerae and Vibrio alginolyticus. Non-sucrose fermenters include Vibrio parahemolyticus and Vibrio mimicus and vulnificus. Okay? So those are the ones that you need to remember. Okay? Now, we have also species of Vibrio that is associated with wound and ear infections. Okay? Species of Vibrio in marine environments. Ay, bakit naging H. pylori? Yung next. Wound infections and marine environments. Please don't forget Vibrio alginolyticus. Okay? Another one. So the answer to this question, alginolyticus. Alginolyticus. Okay? Siya yung mga nakukuha sa sugat. Sa mga sugat. Yung pag nadulas ka sa mga bato. Okay? Now, we're going to move on with H. pylori. Another name for H. pylori is Vibrio alginolyticus. Okay? Previously known as Vibrio alginolyticus. Vibrio species. Next slide, please. Vibrio species associated with gastroenteritis. This includes Vibrio parahemolyticus, and the reason how it causes gastroenteritis is because of the mode of transmission. It's ingestion. Ingestion of what? Ingestion of contaminated seafood. Okay? So you might have a question that asks, that starts with the case analysis. Food poisoning associated with eating contaminated seafoods and mussels. Okay? 2 days after a day of enjoying shrimp and Oh no, maganda yung gumbo yung napauso. Gumbo. Patrick, Mr. Patrick, a patient that was admitted to the to the um to the emergency department with complaints of gastroenteritis after ingesting gumbo from Louisiana. Mga ganong mga kemerut. Gumbo from Louisiana. Grew sucrose fermenters on TCBS with his stool, positive growth with with alkaline peptone water, oxidase positive, kemerut. Okay? So yun, ganun ang mga tanungan ko, class. Okay? Next, we move on to another species of Vibrio. Which species of Vibrio is acquired from marine environments most likely? Septicemia acquired from marine environments is most likely associated with which species of Vibrio, or what species of Vibrio? Vibrio vulnificus. Okay, vulnificus. Now, we also have to talk about, we're gonna come back to Parahemolyticus because I think these slides were mixed up for some sort of reason. We're gonna come back to Vibrio parahemolyticus. So Vibrio parahemolyticus, what do we know about Vibrio parahemolyticus is that it is associated with seafoods and something like that. Okay? Another information that you need to be familiar with when it comes to Vibrio parahemolyticus is about Wagatsuma blood agar. Now, Wagatsuma blood agar is your produces what we call the Kanagawa phenomenon. Okay? So, Kanagawa phenomenon is, I think that is described on the next slide. If it's from a non-human source, it will be non-hemolytic. Okay? That is Kanagawa phenomenon. But if it is from a human source, it will hemolyze the Wagatsuma blood agar. Okay? So, anong pinagsasabi mo, sir? So, for example, because medical microbiology doesn't just include patients in the hospital. We also work as we also work in public health. Okay? So you may not notice it in your work right now, but sometimes there are cases that we have to investigate where an outbreak occurs. And a great example of finding out whether something came from a human source is by demonstrating Kanagawa phenomenon. Okay, so usually the diseases that we talk about comes from skin, contact to skin, infection via inoculation, mga ganon, di ba? But we never talked about the food safety. Diba? Kasi nga, hindi naman yun part ng work natin. Okay? So, some microbiologists or industrial microbiologists actually perform tests on non-human specimens. Like, for example, pagkain or tubig. Diba? Ganon. And Kanagawa phenomenon is an example of what the other branches of microbiology do. Okay? So, using Wagatsuma blood agar plates, you can demonstrate Kanagawa phenomenon. Okay? So, again, just for verification, just for your information, non-human sources, non-hemolytic. Human sources, hemolytic. Okay? What's the blood agar that we use? Wagatsuma blood agar. Okay? Nakita nyo, parang mga Hapon, yung mga mga naka-discover ng mga ganyan. All right, so we have another organism. We have another agar plate. Okay, TTGA. This is telluride taurocolate gelatin agar, and Vibrio cholerae will appear to have cloudy zones of hemolysis. So parang ano siya, parang may clouds. Okay, cloudy zone of hemolysis. Hindi siya yung clear na green or clear na clear. Okay. In this case, para siyang lipemic yung appearance niya kasi nga may gelatin tayo. Okay? Another type of, please don't forget Dane's phenomenon and Dane's medium. Meron ako mga students na nalilito dyan. Okay? Now, Dane's medium is alkaline peptone water, and Vibrio cholerae can survive in this particular water or particular broth after 6 to 8 hours. Okay? Now, we're going to talk about, this one is an addition, kaya nilagyan ko ng C, kasi current. Yun yung ibig sabihin yan. Okay? L-Tor biotypes of Vibrio cholerae produce yellow colonies on TCBS, but turn to what color after extended periods of incubation? L-Tor colors will turn green. L-Tor colonies of Vibrio cholerae or L-Tor biotypes of Vibrio cholerae will turn yellow to green. Okay? After extended periods of incubation. So you might say, oh no, according to our initial wala nagkamali. Okay? Talagang ganun lang siya kasi nga kakaiba nga tong biotype na to. Okay? Now, let's move on to the biochemical tests for the identification of Vibrio cholerae. So you can use nitroso-indo test or nitroso-indole test or the cholera red test, and it has 3 reagents: peptone water, nitrate, concentrated sulfuric acid, and of course, because it's cholera red test, the positive result is red. Now, the principle of the test is the detection of nitroso-indole. Okay? So, clear? Clear tayo dun? Board exam question about this one is that it's which organism is detected by nitroso-indole? Or which organism is detected by which organism is presumptively identified using nitroso-indole test? Or, okay, mga ganon. Kasi hindi nila gagamitin yung cholera red. Pamigay yun yung pag-cholera red. Okay? Ganon yung tinanong sa board exam ng kaklase ko dati. Okay? Kasi magkaibang date kami nag-take. March ako, September siya. Okay? Kasi mas nauna siyang gumraduate. Okay, next. What animal is used in the animal inoculation test for Vibrio cholera? Okay, so we remembered earlier, guinea pigs. Okay? We observed this time, again, another type of phenomenon, which is Pfeiffer's phenomenon. Not to be confused with Pfeiffer's bacilli. Okay? Magkakaiba yan, class. Dale's phenomenon, Dale's medium. Okay? Pfeiffer's bacilli, Pfeiffer's phenomenon. Huwag niyong pagbabalik-balik na rin, class. Gusto ko gumawa na kayo ngayon ng mga scratch, mga ano ninyo, mga board exam review, ano ninyo, para hindi kayo malito. Okay? Now, what test is used to demonstrate the soluble hemolysis of L-Tor biotypes of Vibrio cholerae? We use the Greigs test, and of course, we have to use blood. Okay? Because we're talking about hemolysis, right? Demonstrates soluble hemolysis of L-Tor biotypes of cholera. So we use sheep's blood. This is known as Greigs test. It's not Greg's test, it's Greigs test. And we use sheep's blood. Okay? Now, we move on to Pseudomonas aeromonas species. Pseudomonas aeruginosa tuloy. Aeromonas species. A hemolytic pattern observed in Aeromonas is beta hemolysis. Okay? And like most species of Vibrio, Aeromonas hydrophilia is associated with what habitats? Of course, it's marine habitats. Hydrophilia nga klase. Diba? We have also Aerococcus urinae. I think I did add the Aerococcus urinae. Did I add Aerococcus urinae? Wala. Okay. So, dagdag natin si Aerococcus urinae dito. Common UTI. Common Aeromonas species associated with UTI. Huwag niyo kakalimutan si Aeromonas urinae. Kasi yan na yung mga ano ngayon, mga bago ngayon, si Aeromonas urinae. Okay? So, di ba, madali lang si Aeromonas. Aeromonas hydrophilia. Di ba? Sa dagat. Ay, sa tubig. Water loving. Aeromonas urinae sa wiwi. Okay? And then, Ay, kamali tayo. Patrick, paalala mo sa akin, send ko sa iyo to ha. Kasi may mga edit tayo dito. Ha?
Sige, sir.
Okay. Nasaan na tayo? Okay. Alright. Okay. So, what animal has been reported to transmit Pseudomonas species by wounds? Leeches. So, animal leeches. So, leeches. What is the characteristic flagella of Pseudomonas species? It has a polar flagella, meaning it's monotricous also. Gastroenteritis associated with Pseudomonas resemble that of shigellosis. Dalawa na yung organisms natin na associated with shigellosis class. Ano yung isa? Enterotoxicogenic E. coli class. Okay? Now, one other important organism that you guys need to remember, tinatanong to sa board exam, oxygen preference of Campylobacter. Ito lang maririnig nyo mayroong sinasabing microaerophilic. And what is microaerophilic? Anong percent yung oxygen natin? Oxygen content natin, class. Sige nga, class. Lagay ako ng line. Tapos tingnan natin kung may makakasagot. Nasa na tayo? Bakit nasa flagella ka pa lang? Hoy, bakit nasa flagella ka pa lang? Kanina pa ako next slide. Medyo na.
Parang
Kanina pa ako eh. Patrick, inaanto ka na.
kailangan ko siyang i-present ulit.
Okay. Go lang. Okay. Si Patrick naloloka lang. Okay. Ayan, okay naman na. Gumagalaw naman. Okay. Nakalimutan mo lang gumalaw. Ikaw talaga. Giling-giling. Okay. So, Campylobacter species na tayo. Campylobacter species, oxygen preference muna tayo. We said earlier, it's microaerophilic. Okay? What does microaerophilic mean? Ilang percent ng oxygen dapat? Sige nga. 5 to 8%. Hindi ba 5 to 10%? Grabe naman to si Lilzo. Binitin naman si ano. Binitin naman yung mga mikrobyo. Kawawa naman yung mga mikrobyo. Okay. So, kayo ni Ronchbell, pagsamahin ko kayo na pagbuhuling ko kayo ni Ronchbell. 5 to 10%. Ilan ba ang aerobic conditions? Sige nga, for microorganisms hindi for tao. Baka mamaya may sumagot ng for tao, 100% of course. Ilan ba ang aerobic? Sinabi ko yung last week, nung nag-lecture ako sa inyo last week. Ilan yung bibigyan ko na kayo ng choices. Along ko kayo, mga estudyante ko kayo. Hoy, anong 5 to 14? Ay, nawala tuloy yung mouse ko. Ba't nawala ang mouse ko? Ang mouse ko nawala. Nawala mouse ko. Ano nangyari sa akin? 20%. Jericho naman, niloloko naman yung Jericho. 21%. 20 lang. Kayo talaga. Last na one eh. Okay. 20. Ano ba yan, Jericho? Kayo magbubuhol-buhulin ko kayong apat ha. Ay, kayong tatlo ha. Isama na natin si Patrick. Kahit walang ginagawa si Patrick. Okay. Teka lang, nawala yung mouse ko. Ano nangyari sa mouse ko? Okay. So, next slide, please. Describe the motility pattern of Campylobacter species. Corkscrew motility or dashing motility. O sige, Patrick, paano mo ika-character? Pakita mo sa amin ang corkscrew motility. Para hindi ko talaga ikaw. Ano yan? Bakit may gano'n? Hoy, hoy, hindi ka namin pinagsasayaw, bro. Daliri lang. Ayan, para nangungulamot lang. Visit talaga to si Patrick. Okay. So, hindi parang nangungulamot. Yung iniikot-ikot yung kamay niya, class. Huwag kayong maniwala dyan kay Patrick. Patrick, you lie. You're playing with our time, Patrick. Okay. Next slide tayo. Okay. So, seagull wing appearance. Oopsie. Bakit nawala yung mouse ko? Ano nangyari sa mouse ko? Disconnect ang mouse ko. Tama ba yan? Sandali. Wala yung mouse mo.
Anyways, let's go with next slide. Kasi nakaharang.
Pinapahirapan ako ng computer ko, ha?
Okay.
Alright, so seagull wing appearance, Campylobacter jejuni, and the culture of organism is done on Campi agar or Sciurus media. So hindi kayo mahirapan yan, class, ha? Pag sinabing Campi pack, iyan yung parang pouch na nilalagay natin sa anaerobic jar. Okay, we don't actually use an anaerobic, we don't actually induce an anaerobic environment, but we have a microaerophilic environment with the Campi pack. Okay, next slide. Okay, huwag nga kalimutan. Mode of transmission of Campylobacter jejuni is contaminated, ingestion of contaminated poultry or water, and it has a similar mode of transmission with Salmonella. Again, similar mode of transmission, Salmonella, yada yada. I think you guys know already what we're talking about. Okay, and next slide. Notable biochemical characteristics of Campylobacter species. Huwag niyo kakalimutan yung CHO. Okay, what does CHO stand for? CHO stand, oops, ano ba nangyayari sa akin kung ano mga pinipindot ng ano ko. Okay, so CHO stands for catalase, high purate, utilization, and oxidase test. And everything in there is positive. Okay, so remember, class, it is a close relative of the Vibrionaceae family. Therefore, it will produce an oxidase positive result. Okay? Even though it is microaerophilic. Now, let's talk about the pathologic conditions associated with Campylobacter jejuni. I think that's the next slide, right? Not just Campylobacter jejuni, but also Campylobacter coli. So for Campylobacter jejuni, you have gastroenteritis, diarrhea, and abdominal pain. For Campylobacter fetus, septicemia in newborns, hence the term Campylobacter fetus, and then coli, enteritis. Okay? So please be mindful of those two guys. Okay? Optimum temperature for the growth of Campylobacter species. Optimum temperature for the growth of Campylobacter species is 42 degrees Celsius to 43 degrees Celsius. Yan yung sinasabi ko sa inyo kanina, class. Yung dedicated, may dedicated incubator siya. Diyan kami naglalagay ng Pseudomonas, presumptive Pseudomonas organisms namin. Yung hindi pa namin sure. Okay? Naalala nyo kanina yung pinag-usapan natin yung Pseudomonas, di ba? Pseudomonas aeruginosa is the only one that grows at 42 degrees Celsius. Di ba? Next slide, please. Another one that we need to talk about is Helicobacter species. So, Helicobacter is, again, another one of those organisms related to Vibrionaceae and related to Campylobacter species. Actually, Helicobacter pylori previously was known as Campylobacter pylori. Okay? But because of molecular testing and gene analysis, they found out that it is a separate organism altogether. Okay? So we have pathologic conditions. It causes peptic ulcers, and its former name is H. pylori. Campylobacter pylori. Ulcerations in the stomach caused by Campylobacter jejuni is its ability to convert acid pH to alkaline. This is because of what enzyme? Urease. Okay? It has a urease enzyme. So, siya lang yung organism na may high urease activity. It has such a high urease activity that it can turn your 1.0 or your pH of 1.0 in the stomach to an alkaline condition. Okay? So, yun. Let's go and move on to the next one. The specimen of choice to identify H. pylori is stool or stomach biopsies. But I wrote here in the exam in your what do you call this? Your PowerPoint presentations as biopsies, the best one, because it's asking specimen used to identify H. pylori. But nowadays, because this is such an old, this is such an old, what do you call this? It's such an old presentation. Nasaan ang mouse ko? Ba't nawala? Ayan. We can now use serological testing. Yung chromatography lang naman siya, class. Okay? Ano ba yung chromatography? Parang preg test, pero yung gamit mo is yung poop ng patient. Or you can also use the urea breath test. Okay? So, you'll talk more about that when it comes to analysis of urine and body fluids. Okay? Because those are different tests altogether. Okay? Now, next slide, please. Describe the flagellar structure of H. pylori. Flagellar structure of H. pylori. So, nasaan ako? Nawala yung ano ko. So the flagellar structure of H. pylori has it's a Lopotrichus flagella that has 4 to 6 polar flagellum. Right? All right, and I think that's the end of. Oh no, there's more. Go to the next slide. Hemophilus. Okay. In some countries they spell it like this. In some countries they spell it as hemophilus. Sometimes they put an HA in between H and E. Okay, they put an A there, right? It doesn't matter. It's still the same organism. Okay. Now, oxygen. Let's move on to the next slide, Patrick. Oxygen preference of Hemophilus species include it's a facultative anaerobe, and it grows well at 35 to 37 degrees Celsius. Facultative anaerobe. It does have nutrient requirements, which is the next one in the next slide. It does have nutrient requirements and tests used to differentiate them. You have to test delta ALA or D ALA or X factor or V factor. Okay? Hindi siya factor X at saka factor 5 class. Ay, dito hematology. Okay? May delta ALA din sa hematology. Kaya huwag kayo ma-confuse, class. Iba yung X factor at saka yung V factor at saka delta ALA. There are other names for that one. Okay? So next slide, please. This molecule is the precursor molecule for hemine synthesis. This is delta ALA. Okay? And Hemophilus species that has this are considered to be X factor independent. Okay? So, for example, or hemine independent, for example, if the organism is positive for delta ALA, you will see X factor negative. Okay? So it doesn't need hemine. Okay? And that's the other way around. Okay? If it needs hemine, it doesn't care for delta ALA. Okay? So ganun siya kaarte. Okay, next slide, please. Other names for the factors that is required by Hemophilus. Huwag niyo kakalimutan, class. X factor is hemine or porphyrin. V factor is NAD. Okay? X factor is, of course, heat stable. Let's not forget about that because that's what you see in chocolate blood agar plates. V factor is the one that's heat labile. Okay? So yun lang tatandaan nyo. Pag hemine ay nabulok na yung kaya nagiging chocolate agar kasi nare-release from hemoglobin yung di ba? Kaya siya nagiging chocolate agar kasi nare-release yung hemoglobin from ay, ano ba yan? Okay? Nare-release yung hemoglobin from the red cells. Okay? Alright? NAD, on the other hand, you add this to the agar. Okay? Next. Hemophilus species that are X factor independent. There are 4. Hemophilus parainfluenzae, Hemophilus hemolyticus, Hemophilus aprophyticus, and Hemophilus paraaprophyticus. Napakadaling tandaan, ang daming para. Parainfluenzae, parahemolyticus, aprophyticus, and paraaprophyticus. Di ba napakadaling tandaan, class? Okay? Examples of Hemophilus species that are V factor independent. Ito naman yung mga hindi kailangan. Hemophilus ducreyi and Hemophilus aprophilus. Sir, I have a question. Bakit dalawang beses ko nakita si Hemophilus aprophilus? Magkaiba yung aprophilus at saka aprophyticus, class. Balik mo doon, balik mo two slides above. Okay? Ayan, aprophyticus yung isa. Ito namang nasa baba, pakita natin. Aprophilus. Okay? Aprophilus. Okay? Clear? Clear tayo, class? Huwag mo kakalimutan si Hemophilus ducreyi, class, kasi yan yung important sa atin. Okay, now that we've understood that one, let's talk about the sources of factor X and factor V. Factor X is usually found from degraded hemoglobin, as I've said before. Okay, so when you make chocolate blood agar plates, you already have factor X. Okay, but factor V, on the other hand, is something that you add artificially. Okay, it usually comes from potatoes. It comes from yeasts. Sometimes it comes from some microorganisms. And that's important because there is what we call satellitism. Right? Now, next slide, please. We're going to talk about organisms that produce factor V. Factor V. Okay? Huwag kakalimutan sa pneumococci. Ano ba yung mga pneumococci natin? Streptococcus pneumoniae, di ba? Okay? At saka ano pa? Staph aureus and Neisseria. Look at them. What's the common trend with them? They are all cocci. Di ba? Pansin nyo. Huwag nyo kakalimutan, kasama din pala sa pneumococci is si Moraxella cataralis. Okay? So, ano sir ang importance? We'll go to the next slide. Examples of beta hemolytic hemophilus. You need to remember that particular slide later because we will see why. Okay? It has something to do with satellitism. Okay, so examples of beta hemolytic hemophilus is of course, beta Hemophilus hemolyticus and Hemophilus parahemolyticus. Okay? Another name, and let's go to the next slide. Another name for, I think it has something to do with Hemophilus influenzae. Yeah. So another name for Hemophilus influenzae is Pfeiffer's bacilli. O, di ba class? Huwag nyo kakalimutan si Pfeiffer's phenomenon at saka si Pfeiffer's bacilli. Magkaiba yun. Okay? Tandaan nyo na class. Okay? So Pfeiffer's bacilli is that silent P. Okay? Pfeiffer's bacilli is characterized by dewdrop colonies. Okay? That's the colonial morphology, and again, that's for Hemophilus influenzae. Let's move on to the 6 serotypes of Hemophilus influenzae. So you have 6 serotypes, and it's very easy. A, B, C, D, E, F. Okay, A, B, C, D, E, F. The most commonly encountered is the one that you need to remember, and that is serotype B. Okay? Is this important for us? Yes. Sometimes, especially as in when we do molecular laboratory testing, when we run the Hemophilus influenzae, usually we would isolate this from either a respiratory specimen, a CSF, and then when we run the sample to a molecular array, it will give us options of A, B, C, D, E, and F. Okay? The reason why serotype B is important and it's most frequently encountered is because it's the one that we see in the CSF. Okay? The most common, let's go to the next one. Again, we talked about Hemophilus influenzae before when we were discussing the pathogenic determinants. And what do you remember about it? Is that it has a capsule. Okay? So it has a capsule. That's how it causes the disease. Okay? Specimens that may be used to identify Haemophilus influenzae, as I mentioned before, blood, CSF, pus, and nasopharyngeal swabs. Okay? Nasopharyngeal swabs or NPS. Yun yung NPS, nasopharyngeal swabs. Okay? Clear? Clear pa ba so far? Okay. We keep on moving on. Haemophilus influenzae is the main cause of meningitis in children of what age? This is actually a question that was taken from the ACP Board of Certification question bank. Okay? Of what age? 5 and below. Okay? Huwag kakalimutan 5 and below. Next is what? The next one is stop ka muna dyan. Hindi yan isolate isolate yan. Okay? The next one is Streptococcus pneumoniae, Listeria monocytogenes. Okay? Huwag niyo kakalimutan yung age groups. Ay, hindi pala. Yeah, tama. Haemophilus influenzae, Streptococcus pneumoniae, Listeria monocytogenes, and then next is Neisseria meningitidis. Okay, class? Huwag kakalimutan na. Okay? So, punta tayo sa culture media used to isolate Haemophilus influenzae. We can use BOP and CAP. We can also use Leventhal and Fields and bacitracin BAP or blood agar plates. So, ano naman yung pinakaiba niyan, sir? So, yung bacitracin BAP na yan, meron lang yung dosage ng antibiotic na bacitracin. And that's basically it. Okay? Because many organisms don't survive bacitracin except for Haemophilus influenzae. Okay, now we're going to talk about the next slide, something to do with the colonial characteristics. The odor produced by Haemophilus species is a mousy odor. Ano yung mousy odor na yun, sir? Amoy dagis or amoy daga. Yung amoy ihi ng daga. Sino na lang pag-alaga ng hamster dito or ng dagang costa? Di ba meron siyang kakaibang amoy? Yung parang daga lang talaga. Amoy na daga lang talaga. Ganon. Yun yung amoy niya. Mousy odor. Okay? Next. Nasa si Patrick? Ano nangyari, Patrick? Ba't nawala ka? Mousy odor ka lang, Patrick. Nag-mousy odor lang si Patrick. Nawala na.
Namoy ko, sir.
Ay. Next slide na tayo, Patrick. Ano ba? Boy, you played without time. Okay, what is observed when Haemophilus influenzae is inoculated with a factor V producing organism? Siyempre, yun yung tinatawag nating satellitism. Okay? Okay? So, satellitism. Ano yung satellitism, sir? Imagine nyo si Patrick yung factor V producing organism. And then, sabi niya, lilibre ko kayo Starbucks lahat. O, di nagdikitan kayo lahat kay Patrick. Di ba ganon? Yung mga klase lang natin ng mga richness, katulad ni Lilzo, mga ganon. Yun lang yung hindi natuwa dun sa ano na yun. Sabi, ayoko lang, gusto ko ililibre mo ako sa Mary Grace din. Tapang tita, Mary Grace. Okay, so that's satellitism. So for example, there's one colony is surrounded by one colony of, let's say, Staphylococcus aureus. Inoculated it with the control of Haemophilus influenzae. Okay? So alam natin na Haemophilus influenzae yun. We try to streak it together perpendicularly to that one, and you'll see that you'll see after 24 hours of incubation, tumubo pala, sir. Ganun ang nangyari. Okay? So yun. Clear ba tayo doon, class? Clear? Malapit na matapos tong tong lecture na to. Let's talk about Koch-Weeks bacilli. Okay? Okay, so let's talk about Koch-Weeks bacilli. Hindi gumagana eh. We'll talk about Koch-Weeks bacilli. Hang on a sec. Need to fix something. There's something wrong. Ayan. dito na tayo. Cotchwicks bacilli. Okay. So another name for Cotchwicks bacilli is what? Another name for Cotchwicks bacilli is Haemophilus aegyptus. It has a close resemblance to Haemophilus influenzae. It's also known as biotype 3. Closely resembles Haemophilus influenzae biotype 3. Haemophilus aegyptus. Cotchwicks bacilli is actually important. Next slide. Is important because it is the causative agent for. Hang on a sec. Nasaan na yung ano ko? Slideshow ko na sarili ko. Ayun. Na-stuck siya ng matagal sa microaerophilic. Hello.
Oo, sir. Minsan nagaganin ko eh.
Pati yung akin, naloka ako. Okay. Pati yung akin. Okay, so wala tayong magagawa doon. Class, at least meron tayong babalikan natin siya class ulit. Pero charit lang. Ay, hindi, charit lang. Nakakaloka kayo, walang ganun. Meron naman akong lecture na iba dyan. Hanapin nyo na lang pagka naloka kayo. Or sabayan nyo na lang whilst you're listening. Okay, ganun tayo dito, class. Sometimes may mga nangyayaring kaguluhan. Okay? Kaya pala, ito si, hindi nagsalita. Sir, ano nangyari? Sana man lang na ganun ka. Ikaw talaga. Okay, sana ako. Medyo madami pa pala siya. Hindi pala onte. Medyo madami. Oops. So, mag-stop tayo with, mamaya mag-stop tayo later. Okay? So, Cotchwicks bacilli na tayo, di ba? Okay. So, Cotchwicks bacilli is the causative agent of pink eye. Okay? Ayoko magkwento ng kalokohan. Isasabi ko na lang i-chat ko na lang kay Patrick later kung saan yung makukuha yung pink eye. Pero basically, pink eye is sore eyes, class. Okay? Mamaya ko sabihin kay Patrick. Walang kahalong kabastusan yun obviously hindi naman niyo ako professor, so I have no liability towards that one. Pero, just in case, kasi public health, professional din ako, di ba? Patrick, sabihin ko sa'yo kung bakit. Mamaya, tapos sasabihin nyo sa mga kaklase mo, ha? Okay, kasi malay nyo may makuha kayong case presentation na ganyan sa exams ninyo. Okay? Pero I think may idea si Patrick kung saan nakukuha yung pink eye.
Ay, sir, wala po.
Wala kang idea, malinis. O sige, sisirain ko yung buhay ni Patrick talaga. Okay, anyways. So, Haemophilus influenzae, Haemophilus ducreyi is the causative agent of chancroid or soft chancre. Okay? And it is the smallest pathologic bacteria. Okay? Alright. And characteristic appearance of that one, when you look at it in the microscopy, next slide tayo, Patrick, sorry, hindi ko pala. Excited ako mag-discuss. Minamadali ko yung mga pangyayari. Next slide. Patrick, the appearance of Haemophilus ducreyi when examined through direct microscopy is school of redfish. Okay? Iba yung fish in a stream tsaka yung school of redfish. Kasi may mga ganong keme. Okay? Now, let's move on to the next gram-negative organism that is not Enterobacteriaceae. It's now Bordetella species. Okay? Most species of Bordetella requires this substance for growth. I think that's our next slide. Okay, so most species of Bordetella requires this substance for growth. It's nicotinic acid. Okay, nicotinic acid. How do I remember that one, sir? Bordetella mostly is associated with respiratory diseases. And wala akong maisip. Pag sinabi kong nicotine, sigarilyo. Hindi naman sigarilyo. Hindi ko sinabing sigarilyo. Mahilig ka mag-sigarilyo. Bordetella, sakit mo. Pero yun lang yung naiisip ko kaagad. Nicotinic acid. Okay? Now, all members of Bordetella are non-motile except for Bordetella bronchoseptica. That is our next slide. The oxygen preference of Bordetella is, of course, it's an obligate aerobe. Of course. Okay? Bordetella species. Obligate aerobe siya, so it requires oxygen. 24/7. Diba? Ang arte. Or ambient. Ang arte kasi ng mga terminologies ng mga katrabaho ko dito. Ambient temperature daw. High ambient conditions. Ganon. Another name for Bordetella pertussis or pertussis is Bordet Genju bacilli. Okay? And it is the causative agent of whooping cough or pertussis. Okay? Dito ko lang nalaman na ang basa pala niyan, hindi pertussis. Okay? Pinoy na Pinoy yung basa natin o pertussis. Para maiwasan ang pertussis, kailangan natin kailangan bakunahan ng mga bata. Di ba? Pertussis ang basa natin. Dito pala sa Amerika, ang basa nila din pertussis. Ang arte, di ba? Okay? Now, what is the ideal specimen for the isolation of Bordetella pertussis? It's actually nasopharyngeal swabs, okay, or NPS. Before, it was known we have cough plates, kaso madaming nahahawa, kaya nasopharyngeal swabs na lang. Okay? Next, pathologic determinants of Bordetella pertussis or pertussis. We have endotoxin, exotoxin, and hemagglutinin. Okay? Now, we're going to talk about the disease or the stages of pertussis in order. It starts with the catarrhal stage, paroxysmal stage, and then ends with the convalescent stage. It's one of those diseases that that when you recover, you become much more infectious. Okay? So, we start with the catarrhal stage. The patient is asymptomatic but highly infectious. Okay? The paroxysmal stage is where you see the inspiratory woof. Yun yung gumaganon. May mga bata na ganon. Di ba? Ang ganda ng sound effects, Patrick. Naloka ka, Patrick. Gumaganon. Yun yung mga bata na may ano. Okay? Lalo na yung mga hindi nabakunahan. Okay? May ganon ka din, Patrick. Ba't ka nagtaas ng kamay, Patrick?
Inform ko lang po kayo. Check ko po kasi yung live stream, pero parang naka-gray out ng yung presentation.
Naka-gray out siya.
Yes po.
Totoo.
Yan!
Totoo ba? Okay na. Bumalik na. Totoo ba, Patrick? Pinaglololo ko kasi ako nung ano eh. Okay, so there are 3 stages. Don't forget. Okay pa rin, Patrick, ha?
Yes, sir. Okay po.
Okay. So there are 3 stages: catarrhal, paroxysmal, and convalescent. Okay, 5 convalescent stages, basically the recovery stage. Okay? Clear pa tayo, guys? Nakakasunod pa. Hindi pa naloloka, hindi pa inaantok. Kasi tuloy-tuloy na natin ito hanggang mycobacterium. Charet. Charet, class, charet. Magsi-second break pa tayo, class. Chill lang kayo dyan. Okay? Bakit ganun? Oo nga, naka-gray out nga.
Oh, sir, nagbalik siya ng gray out. Pati yung mga video po nung
No?
mga
Nag-gray out siya. Oh my God. Talagang, ayaw na talaga niya tayo mag-ano. Ayan. Screen capture. This one. This one. This one. Slideshow. Ayan. Ano natin? Okay. Yung mukha ko lang yung nakikita na gumagalaw. No?
sir, ay lang gumagalaw.
Oo. Ayan, okay na siya. Nakita ko na naman yun. Okay. Buti in-inform mo ako, Patrick. Okay? So, next. Next thing that we need to talk about is of course, the media used to isolate Bordetella pertussis. Okay? So there's 2 media that we need: Bordet Genju bacilla, Bordet Genju media and the Regan-Low media. Both of them have a different formulations. Sorry, na distract. Have different formulations. Bordet Genju has potato extract and 50% blood and glycerol, while Regan-Low has charcoal, cephalexin, and horse blood. Both of them have antibiotics, but what we want to remember is that it's based on manufacturer preferences. So sometimes they would add cephalexin, sometimes they would add amphotericin. What's the purpose? It is to remove the other organisms that may cause confusion with the diagnosis. Right? So yun lang yun, class. Talaga, sir. Oo, yun lang yun. Para maalis. Kasi usually, ang sample natin is respiratory. Anong problema sa respiratory? Kailangan nating hanapin kung sino talaga yung nagkukos ng sakit ng pasyente. Yun yung trials and tribulations ng mga tao na nag-work sa microbiology. Sino ba ang dapat kong i-work up? Kaya yan, kailangan natin siya ng ganun. Okay? Next. Let's move on to the next one. The characteristic appearance of the colonies of Bordetella pertussis. My gosh, class. Pag ito nakalimutan yung class, ay naku. Ano class ang sagot? Sino ang makakasagot? Everybody in the chat. Paki-chat nga, class. What is the characteristic appearance of the colonies of Bordetella pertussis? Napaka-easy niyan, class. Ang hindi makasagot, quiet na lang ako. Ganun. May sumpa pa rin, pero quiet na lang ako. Ano sagot dyan, class? Who knows the answer? Mercury droplets. Very good, Jericho. Si Jericho ang nakasagot. Mercury droplets. Very good. That is the correct answer. Or pearl-like colonies. Pwedeng mercury droplets, pearl-like colonies. Okay? Okay na yun. Hindi masyadong nag-aano si Sir Manuel. Kasi hindi yan most of the time hindi yan tinatanong. They will show you a picture. Okay? Kennel cough is caused by what species of Bordetella pertussis? It's Bordetella bronchoseptica. Okay? Let's not forget about Bordetella bronchoseptica. What tests are used to identify Bordetella species? You have a bunch of tests. You start first with urease. You then go with motility, nitrate, and oxidase. Okay? So class, paano namin maalala to? UMNO. Huwag niyo kakalimutan yung UMNO. Ano yung UMNO, sir? UMNO. Okay? Urease, motility, nitrate, and oxidase. Now, sir, paano namin maalala si Bordetella pertussis? Negative siya for urease and motility. Okay? Negative siya for urease and motility. It's positive with nitrate and oxidase. Okay? It's positive with nitrate and oxidase. Usually, they would ask about Bordetella pertussis most of the time. Okay? UMNO. Huwag kakalimutan yung UMNO. Okay? Umuwi ng hindi ko maalala. Basta mayroong sinabi yung kaibigan ko dyan basta ganun. Anyways, let's talk about Bordetella parapertussis. So, parapertussis, negative siya with everything except for urease. And then, Bordetella bronchoseptica, motility and ano lang siya, oxidase. Okay? Motility and oxidase. Okay? Clear? Let's move on with Francisella tularensis. Francisella tularensis is the disease caused by Francisella tularensis is tularemia, obviously, or also known as rabbit fever. Bakit rabbit fever? Kasi dati nung nag-experiment ang mga old or mga ancestors natin, ng mga microbiologists, ancestors talaga ang term ko. Yung mga rabbit na yung mga rabbit experimental animals nila sa lab. Yun yung nag-cause ng fever sa kanila. Okay? Yun yung nag-cause ng sakit sa kanila, class. Okay? So actually, if you have Francisella tularensis growing in the laboratory, it has to be reported to the Infectious Control Committee. Kasi ibibigyan ka ng prophylactic antibiotics. Okay? Now, we have mode of transmission for tularemia. Obviously, direct contact, ingestion, and arthropod bites. So, any organism that bites an infected organism, like for example, somebody who got tularemia. Let's say Patrick got tularemia. Okay? May maraming garapata dun sa bahay ni... I'm not saying now, ha. Theoretically lang to, Patrick, ha. Hindi ko kinukutya ang estado mo sa buhay. Alam kong richness ka, Patrick. Okay? So, let's say for example, yung lugar na, yung dorm na lang ni Patrick. Kaya, ganun. Baka kasi sabihin nung pamilya ni Patrick na habang narinig niya ako nagle-lecture, kinukutya ko sila. Ano na lang. Theoretically lang to, ha, class, again. Okay, so si Patrick nagkaroon ng tularemia, nag-work siya for example sa PGH. May patient doon na nag-positive and then hindi na i-report na meron palang tularemia ang patient. So nadala ni Patrick sa may dorm niya. Okay? Eh, dun sa dorm niya, madaming garapata. Okay? Pwede ka pa ding mahawa nun. Okay? Pwede ka makakuha ng tularemia dun sa garapata na nakakagat kay Patrick. So, for example, yung garapata na yun, mga garapata na nakakagat kay Patrick ng itlog, tapos infected na din yung nanay ng mga garapata na yun. Okay? Na i-transfer dun sa aso. So, ganun ang pagkalat ng tularemia. Masyadong mabilis. Okay? Kaya kailangan i-report sa Infection Control Committee. Pagka nandito yan sa amin, sa laboratory namin, tawag ka, naluluka na kaagad yung mga infectious disease specialists namin at saka mga fellows. Naluluka na sila. Pupuntahan na sila doon. Kahit na nakakahawa yan, titignan nila yan. Kasi very rare lang yun na occurrence. Okay, so direct contact, inhalation, ingestion, and arthropod bites. Imagine, even the laboratory personnel could get infected. Okay? So, let's start first with the antibiotics that is usually incorporated in culture media for Francisella tularensis. Usually, we add penicillin and cyclohexamide. Okay? Penicillin and cyclohexamide. The media of choice for Francisella tularensis is glucocysteine blood agar. Other media includes PCA or CHA. Okay? Charcoal hemagglutinin agar. Okay? Potato. Oh no, it's not potato. Penicillin pala, sorry. Penicillin cyclohexamide agar. Sorry. PCA is penicillin cyclohexamide agar. Alright, next, we move on to Brucella species. Brucella species, what you need to remember is it causes what we call undulant fever or Malta fever. Undulant fever or Malta fever. Anong ibig sabihin ng undulant? Patrick, gagamitin ko ulit ikaw as an example. Ay, iba naman, si Lil Zo. Nasa si Lil Zo? Ayan, Lil Zo, patingin na mga kamay mo, itaas ang kamay. Okay. Yan. Now, go sideways. Isang kamay. Left hand. Sideways yung isang kamay. Yan. Can you mimic a tidal wave or a tsunami? What does it look like? Can you mimic a tidal wave or tsunami? Ganun ka, wave. Ganun, wave ka. Hindi makita kasi si Patrick nga. Patrick, pakita mo sa amin ng ang tsunami. Ganun. Yung wave ba? Wave. Ganun. Tsunami. Patay tayo with dyslexia to. Ayan, ganyan. Ayan. Yan yung undulant fever. Bakit sa tingin yung undulant fever ang tawag? Kasi may rise and fall. Okay? Tumataas, bumababa, tumataas yung temperature ng pasyente. Kaya tinatawag siyang undulant fever. Okay? So pag ang case presentation in your class, patient was admitted to the male ward with an erratic fever. With a fever that has with an inconsistent fever for almost 7 days. Inconsistent fever, dynamic fever, mga ganun na mga cases yung paiba-iba. Tandaan nyo na si Francisella si Bordetella pertussis. Sa Bordetella tuloy. Pati ako inaantok na. Si Brucella. Tandaan nyo si Brucella. Okay? An additional information for that one is, of course, yung trabaho ng pasyente. Tatandaan nyo yung trabaho ng pasyente mamaya. Mamaya ako sasabihin sa inyo kung bakit. Okay? Now, next slide na tayo. Okay? So, huwag nyo kakalimutan yung kamay ni Patrick na medyo parang parang ewan. Okay, class? So, what species of Bordetella is considered to be normal flora of the gut, GIT? Or animals rather. So, Brucella abortus is from cattle. Brucella miletensis is for sheep and goat. Brucella suis, pigs, and then Brucella canis is for dogs. So, bakit pinaalala ko sa inyo class ang occupation ng patient? So, for example, on and off fever, a case is patient was admitted to the male ward, fever of unknown origin with on and off spikes. Okay? On and off spikes. He works as a shepherd. Mga ganun ang trabaho. Okay? Kung yan yung isa sa mga tandaan ninyo. Pag binigyan na kayo ng case presentation na in-include yung trabaho ng pasyente, tandaan nyo, pagka may naalala kayo, sheep, goat, pig, and dogs, or basta shepherd ang pasyente, most of the time, ang pinapaintindi sa inyo or ang pinapahanap sa inyo niyan is bordetella. Brucella. Brucella. Okay? Brucella ang pinapahanap sa inyo. Kaya huwag kayong mag-alala. Napakadali lang. Kailangan nyo lang hanapin yung mga keywords. Sinasabi ko sa inyo. Okay? Another term for Bang bacillus is Brucella abortus. Okay? Bang bacillus. Okay? Walang kabastusan. Nakangisi ka, Patrick. Nakikita ko. Tsaka kayo dalawa ni Moyo. Puro kayo kalokohan talaga, ha? Yung mga mata ninyo ni Moyo tsaka ni Patrick. Ako, nakikita ko kayo dito, ha? Okay, culture of choice for Brucella. Nagulat si Moyo. Ano kinalaman ko dyan? Okay, so culture media of choice for Brucella is, of course, the Castañeda broth or Castañeda milk broth. Sometimes they just call it Castañeda broth. The organism can survive in this particular media for 3 to 4 weeks. Okay? Aside from the media of choice, what other media may be used to isolate Brucella? So you can use Brucella agar or the Wisconsin media. Very rarely do we use this because this is very old. The Castañeda na usually yung ginagamit. Okay? And because of the advancements in technology, in medical technology, actually, yung blood agar culture bottles, yung blood culture bottles natin, pwede na siyang palitan to 21 days. Pag sinabing 21 days na ang aerobic at anaerobic media natin for blood cultures, the doctors are also thinking that it could be Brucella. Okay? So may mga ganon. So wag kakalimutan, class. Blood culture natin ngayon medyo malupit na. Gumagamit na din tayo ng Meron nga actually mga blood culture bottles na designed for Bactec ang pinagsabi ko, ha. Ganito, yung Bactec TM. This is not a paid promotion. Baka sabihin ninyo, this is not a paid promotion, but if Bactec or BD wants me to work for them to do this every time, I would. But Bactec bottles or the BD bottles have a specific bottle with a serial number that only processes a sample or that has a a protocol for for Brucella lang. So 21 days siya, and then what it does is that what it has inside it is basically the Castañeda formulation. So yun. Okay? So, wag kalimutan yan. So, Castañeda broth, Castañeda media, which is basically a milk broth, 3 to 4 weeks. Okay? Aside from that, we can use Brucella agar or Wisconsin media. Okay? Now, we want to talk about brucellosis some more. Mode of transmission of brucellosis is direct contact with infected products, ingestion of infected animal products, and inhalation. When I was in Saudi Arabia, I was surprised. You could get brucellosis from camels. Okay? I didn't know that. You could get Brucella militensis. Okay? You could get Brucella militensis actually from camels. Okay? But that's not included in your board exams. What we want to focus on is what's in your board exams. Okay? 21 days yung dapat yung tandaan. So direct contact with infected products like milk, okay? Like the meats. Okay? Kaya kailangan, pag tandaan nyo dapat yung occupation. Pag in-inform kayo sa case study na kung ano ang occupation ng pasyente, there is a 75% chance Brucella ang tinatanong. Okay? So clear? Alright. Next. Tests used to identify Brucella species. So there are 2 tests to differentiate them. CO2, growth in CO2, of course, and H2S production. So among the other gram-negatives that is not Enterobacterialis, H2S production is observed in Brucella species. So Brucella abortus will test positive for H2S production after 4 days, and then Brucella suis, the one that comes from pigs, is 5 days. Okay? Clear? Everything else will test negative. And then when we look at CO2, it's only Brucella abortus that will test positive, that will grow at CO2 with CO2 enrichment. I mean, okay. There are other things that you can do to differentiate the species of Brucella. Brucella can be grown in milk media with threonine and basic fusion. Okay? Threonine and basic fusion. And organisms that can grow on threonine is Melitensis, Brucella melitensis, Brucella canis, and Brucella suis. The ones that grow in in basic fusion is Brucella abortus and Brucella melitensis. Okay? Now, let's move on to the skin testing. Skin testing, we test for what may be used for the skin testing, which it's basically an antigen, an antigenic preparation of known as brucellin or abortin. Okay, so if it's brucellin, it's generally the mixture of all. It's generally like a choice of Brucella abortus, suis, melitensis, and canis. If it's abortin, it's basically just Brucella abortus. Now we have again another one. Next slide. Milk media test for Brucella. So we have two tests for milk. We have two tests for milk media. We have two reagents for the milk media test. Another name for the milk media test, by the way, is the abortus bang ring probe test, which requires 1 ml of milk and 1 drop of hematoxylin stained organism. The positive result for this one is a violet ring. Again, this is obsolete, but what I want you guys to remember is that if you have the Castañeda milk broth, you can do this test immediately. Okay? And all you need to do is to just make a bacterial suspension with hematoxylin, and then drop it in the Castañeda milk broth. You'll see a violet ring on top. Did we do it on my day? No, we don't do it. We don't do it anymore because you just put it in the Malditoff machine. Okay? Pasteurella multicida was previously known as Flavibacterium multicida. It is contracted from. Oh yeah, we're going deeper into bacteriology. By the way, Pasteurella multicida is usually contracted from two animal vectors, which is either cats or dogs, because these organisms are the normal flora are the oral cavity of the normal they are the normal flora of the oral cavity for cats and dogs. So you'll get rat bite fever from that one. Notable biocharacteristics of Pasteurella multicida is that it is oxidase positive and indole positive. It's very rare, or it's very unusual for something to test positive with oxidase and indole positive, with indole also, right? Most of the time, most of the organisms that we talked about earlier are just indole positive, but rarely would you hear it as oxidase positive as well, right? Earlier. So ano bang indole positive? Mostly Enterobacteriaceae or Enterobacterales, 'di ba? Diba? So we're moving closer to the harder parts of bacteriology na class. Okay? Sugars fermented by by Pasteurella is glucose and sucrose. Okay? And next slide, we have a hemorrhagic and septicemic or hemorrhagic form of septicemia of cattle associated with Pasteurella multicida. This is known as shipping fever. Bakit shipping fever? Kasi usually that type of fever, if the organism is infected whilst it's shipped, or that was the original terminology for it. Majority of the cases, the cattle dies during the shipping process. Kasi siniship sila class yung mga cattle. Okay? So, namamatay yung mga ano. That was the original name. Okay? Now, Pasteurella multicida, next slide. The Pasteurella multicida is associated with what type of infection? This is wound infection from animal bites. Kasi kanina nga, sabi ko kanina, di ba? Oral cavity normal flora siya ng aso at mga pusa. Di ba? Scratches also. Di ba? Kasi yung mga yan, dinidilaan nila yung sarili nila, class. Di ba? So, very, very careful. So, if you encounter a case presentation wherein there is an animal bite, make sure it is cat or a dog. If it's a reptile or anything that comes from what do you call this? That comes from the aquarium. Ano nga yung sinasabi ko kanina? Edward, Ciela, di ba? Hindi na kayo nahihirapan, madali lang yan, class. Just maning-mani lang sa inyo yung microbiology. Maning-mani lang ba sa iyo, ano, Patrick? Maning-mani?
Hindi naman mapagis.
Parang iyak na iyak. Kasi Lilzo, maning-mani na lang sa iyo. Di ba binibigyan ko na kayo ng mga teknikan, ha? Lilzo, ikaw?
Malaking tulong yung mga tips.
Oh, mga tips ahoy, ha? O, Lilzo heart lang. Char ka lang, Lilzo. What about Italia? Ay, busy, nanonood ng Tulfo to si Alessandra. Isang heart nga kung natutuwa ka ba doon sa mga tips ahoy natin. Kasi binibigyan ko na kayo ng ng mga parang dapat yung isipin, ay, sinabi ni Sir na kinagat ng pusa. Ganon, kinagat ng ganito. Alam nyo na kaagad dapat yun. Okay? Let's go with Legionella. Okay? Legionella na ba yung ilagay mo? Legionella. Legionella naman, madali lang din siyang alalahanin kasi there are pathologic conditions associated with each species. Okay? Let's start with Legionella pneumophilia. Legionnaire's disease. Okay? Legionella micdae, it's Pittsburgh pneumonia. Yan nasa Pittsburgh. Okay? Legionella bosemani is the Weigas agent of pneumonia, or you call it Weigas pneumonia. Okay? So Legionnaire's, Pittsburgh and Weiga. Okay? The milder form of Legionnaire's disease is what you call Pontiac fever. Okay. Next slide. The media of choice for Legionella pneumophilia is BCYE. Actually, klase nagpunta lang ako dito sa US. Doon ko lang nakita yung actual na itsura niya. Yung appearance na pinagsasabi niya na ground glass appearance. Okay? Pag sinabing ground glass appearance, class, okay, literal na parang dinikdik na glass. Totoo, hindi ko yan na-encounter nung nandito ako, nung nandiyan ako sa Pilipinas, nung ako sa Saudi at sa Pilipinas. Even though tinetest namin yung Legionella na yan. Hindi ko yan nakita kasi wala naman kaming control nun. Nung nakita ko yung control ng Legionella, ay, parang durog na talaga na durog na bubog. Okay? Ganun yung itsura niya. Pagka narinig niyo yung ground glass appearance na yan, class, tapos BCYE agar, alam niyo na dapat yan Legionella yan, class, ha? Okay? Say, for example, it was from a bronchoalveolar lavage. Okay? So, bronchoalveolar lavage or BAL, yung sample, and then it grew colonies on buffer charcoal yeast agar with the characteristic ground glass appearance. Almost make sure, it's almost certain, huwag na kayo tingnan yung ibang choices. Legionella pneumophilia na yung hanapin ninyo. Okay? Tapos in-include pa yung mga respiratory symptoms. Hindi kayo mga doktor. Huwag na nating alamin yan, class. Hindi ko din yan inalala naman. Yung mga class ito, bigyan ko kayo tips ahoy. Huwag niyong pansinin masyado yung symptoms ng pasyente. Ang tingnan nyo yung mga test. Dahil ano ba tayo pagkatapos natin dito? At the end of the day, medtech tayo. Huwag tayong bida-bida. Charet. Huwag tayong bida-bida. May anemia ang pasyente. Ano kaya ang sakit? Che, tumahimik ka. Tingnan mo ang resulta ng biochemicals. Doon ka tumingin. Tsaka kung anong agar tumubo. Binibigyan ko na kayo ng mga terminologies. Oh, by the way, you can also use Philly Gorman agar pala. I forgot. Brown colony siya. Pero ang mas popular kasi is yung BCYE. Okay? Right. Next. Let's move on to the next one. The best specimen for the diagnosis of Legionella pneumophilia is, uy, kumukulog dito ha. Biopsy material. Because we usually we want fluorescent antibody studies, but the specimen that is usually submitted to the laboratory is respiratory specimens like bronchoalveolar lavage, sputum, what else? Ano pa ba? Nasopharyngeal swabs, mga ganon. Okay, so clear. It could be a plethora of microbial tests. Now let's skip this one because I've already mentioned about this. I've already mentioned to you guys about this. Calimacobacterium granulomatis. I'm going to change this one to Klebsiella granulomatis. Okay? Kasi yan yung sinabi ko sa inyo kanina. Klebsiella na siya klasa. Hindi ko siya na-update. Okay? Ay, pala reclassified. Nandiyan na pala sa ibaba. Reclassified as Klebsiella. Okay? So it was previously known as Calimacobacterium. It's now the constitutive agent of granuloma inguinale or donovanosis. The characteristic morphology of patients with donovanosis is donovan bodies. Ano ba yung donovan bodies na yan, Bas? Yan yung maraming, hindi siya bacterial vaginosis. Okay? Because this is, for bacterial vaginosis, what you want to see is Gardnerella vaginalis. And Gardnerella vaginalis is gram-negative. Usually is, it's going to have clue cells. What's different with granuloma inguinale is that the white blood cells is the one that's infected, not the one for bacterial vaginosis like Gardnerella. Gardnerella parang epithelial cells yung pinapalibutan niya. Okay? So si Calimacobacterium or Klebsiella granulomatis, ang tinatambayan niya, white blood cells. Yun yung nakikita sa pap smear na Donovan bodies. Okay? So it's a mononuclear cell with a lot of gram-negative bacilli. Okay? So that's it. Describe a Donovan body. A mononuclear cell or a white blood cell that has a lot of gram-negative bacilli. It's not with a luma kasi ito na slide. Okay? It's with a lot of gram-negative bacilli. Okay? The proposed name for next slide. The proposed name for that one is Klebsiella granulomatis. This is based on the basis of DNA hybridization assays. Okay? Let's move on to the next one. Inabot ba tayo ng hanggang Gardnerella vaginalis? Ayan, Gardnerella vaginalis tayo. Okay? So G. vaginalis or Gardnerella vaginalis is the causative agent of bacterial vaginosis. Nowadays, they don't allow it to grow anymore because it could be tested with serology and it could with vaginal swabs could be tested with serological assay like chromatographic tests yung para sa PEG test. Or we could do PCR test with the Cepheid na lang. Are you guys familiar with Cepheid? Or yung, ano nga bang pangalan yan dyan sa Pilipinas, sa Cepheid na yan? Sino na nagtrabaho dito nung, may mga kaklase kayo nagtrabaho na dati as, smears and mga tawag doon yung nangungolekta ng samples. Swabbers, ayun. Anong ginagamit nilang machine sa laboratory? Kalimutan ko yung ginagamit. Cepheid kasi yun ng family ng Cepheid. Sino nakakaalam ng machine na yun? Diyan sa Pilipinas ko yung nakita. Ay, hindi yung mano-mano. Hoy, binilhan kayo ng DOH. Kayo nakakaloka talaga kayo. Grabe kayo sa DOH, ha? Binilhan kayo ng machine. Hoy, kayo talaga. Ano yun yung ipinapasok mo lang yung cartridge? Meron dyan yun sa Pilipinas eh. Baka alam ni Sir Marco yun. Kasi ginagamit namin yun dito pag-test ng bacterial vaginosis. PCR din yun. PCR na lang namin yan ngayon. Combo meal pa yan ngayon. Kasi kasama na yung mycoplasma, kasama na yung Candida albicans. Ganun, combo meal yun eh. Nakalimutan ko yung pangalan, babalikan ko mamaya. Infinity. Ayun. Infinity ang pangalan. Naalala ko na. Okay? Cartridge lang siya na ganyan. Gagawa ka lang ng master mix mo from the from the vaginal swab. And then yun na yun. Dati-rati hirap na hirap kami niyan kasi may isang bench yan. Dati-rati may bench pa yan na sarili. Nung nasa Saudi nga ako niya, may sarili may sariling time kami niyan kung kailan namin ginagawa yung vaginal swabs na yan. Kasi nga, iba yung pagbasa ng plates niyan kaysa sa other samples namin. Okay? So, ganon. Okay? So, another name for that one, previous names for that one before is Haemophilus vaginalis. Then it got characterized as Corynebacterium vaginalis. Okay? So, let's move on to the next one. Saan na ba ako? Wala na naman yung mouse ko. Okay pa yung natin, live stream natin. Tatapusin lang natin ito, then we'll rest for a couple of minutes. 10-15 minutes, and then we'll go again to Mycobacterium. Right? All right. So describe the vaginal discharge of a patient with Gardnerella vaginalis infection, foul-smelling vaginal discharge, or fishy grayish vaginal discharge. It is true. Correct naman talaga siya, class. Kasi yun. Okay? Kasi naabutan ko yan eh. Okay? So the cytological test for identification of Gardnerella is through pap smear, and we see clue cells. What are clue cells? Basically, they're epithelial cells that are surrounded by gram-negative organisms. Okay? By gram-negative organisms. Okay? The test used to identify the odor produced by bacterial vaginosis is the whiff and sniff test. We use KOS, class. Inaamoy. Huwag kayong maloka, inaamoy talaga yung class. And it is still part of the literature. Whiff and sniff test with the use of 10% potassium hydroxide. The positive result is, of course, a fishy odor. Okay? So, mangangamoy siya when you add it. The isolation of Gardnerella vaginalis, if your laboratory still wants to do so, is using human blood bilayer twin media, or the HBT media. So pag narinig yung twin media, it could be for Mycobacterium tuberculosis or Mycobacteria in general, or it could be used for bacterial vaginosis. Okay? But we rarely use this anymore kasi nga pini-PCR lang siya. Okay? May mga combo meal pa yan. Vag PCR ang pangalan. Okay? Kasama na din pala doon si Trichomonas. Diba? Ganun kasosyal na ngayon ng technology natin. Anyways, let's move on. Tapos ang nakakatawa ng class, high school students na lang yung mga nagra-run. Yung mga lab assistant na lang namin ng mga nagra-run. Okay? Diyan sa atin sa Pilipinas, underpaid na nga tayo. Charot ayoko na politics. Charot ayoko ng politika. Okay, ayoko niyan. Okay, so let's move on with Streptobacillus moniliformis. Streptobacillus moniliformis causes two types of diseases. It can cause Haverhill disease or Haverhill fever, or what we call rat bite fever. If it comes from rats, obviously it's called rat bite fever. If it's Haverhill fever, it's from contaminated milk products. Naihian ng mga daga. Okay? Or natapakan ng mga daga. Or ininuman ng mga daga. Ganon. What special indication is needed when culturing Streptobacillus moniliformis? SPS is toxic to Streptobacillus moniliformis. Ano ba yung SPS na yan, class? SPS stands for what's SPS? Sino nakakaalala niyan? SPS. Class. SPS is next slide na SPS is toxic to Streptobacillus moniliformis. What is the what tube contains SPS ba? Kaya
Yellow po.
Yellow, very good. Yung mga olden times na mga blood culture bottles. But Amanda, what does
Yes, sir.
SPS stand for ba?
Hindi ko po maalala, sir. Baka mabulol lang ako.
Gusto ko marinig. Nakakainis naman to si Amanda. Sodium polyanethol.
Sodium
Yung polyanethol.
sulfonate.
Hoy, chill
Yung
ka
poly
lang.
nakalimutan ko.
Polyanethol. Sulfonate.
Basta sodium and sulfonate yung dalawang S, yung
Polyanethol.
Sabi ko lang, bubulol ako.
Sodium polyanethol. Oh, ayan.
Opo, yun. Patrick, ba't ka tumatawa?
Patrick, anong klase yan? Manda ka sa amin mamaya. Huwag ka mag-alala, girl. I got your back. Bubulihin natin
Thank you, sir.
yan. Huwag ka mag-alala. Bubulihin natin yan. Charet. Saan na ba tayo? Okay. Nawala ako ang aking slideshow sa... Very good, Amanda. Okay, so SPS is found in the old versions of blood culture bottles. Okay? Kasi now it's broth media, by the way. Describe the appearance of Streptobacillus moniliformis. That's our next slide, I think. On thioglycolate, it usually has puffballs or a string of pearls. Okay? And I think the next organism is Capnocytophaga. Okay, Capnocytophaga, what you guys need to remember about Capnocytophaga is the type of motility. Type of motility is gliding motility. Patrick, paano yung gliding motility? Huwag kang gigiling, Patrick. Sasampalin kita, Patrick. Gliding motility. Sige, gliding. Hoy, hindi. Kakainis to si Patrick. Hoy, Lil Zoe, yan yung sinasabi ko sa'yo kanina. Yung gusto kong pinagagawa sa'yo kanina. Yan yung para sa udulant fever. Gliding motility mo, yo. Pakita mo nga sa aming gliding motility. Nahihiya si Moyo. Nakakalok. Gliding motility, yung isa lang yung direction niya, hindi yung gumagalaw-galaw na ganyan para kayo nagkukumpas. Isa lang yung yung direction niya, tapos biglang nagsuslow down siya. Kasi di ba nag-glide ka, di ba? Ano bang itsura mo, Lilzo, nung nag-ice skating ka? Baka sa California ka ba nanggaling? Walang ice skating sa California. Saan ka nanggaling, Lilzo?
Sa Chicago po.
May snow doon, kaya tumigil ka. So, paano itsura mo kapag ka nag-ice skate ka? Di ba dumadahan-dahan kapag magbe-break ka? So, mabilis, then dadahan-dahan. O sige, Patrick, pakita mo sa amin. Pakita mo sa amin, Patrick, kung paano yung gliding motility. Yung mabilis, then dadahan-dahan. Hindi. Bakit may flare? Ano kabadingan yan? Bakit may flare? Ayan, yung daliri mo lang. Yan, ganyan lang. O, ganun. Whole body daw, di ba? Okay, so yan. Imagine niyo yung mga skaters natin, yung mga nag-ice skating. Di ba bago sila mag-slow down, mabilis yung acceleration nila, tapos bigla sila magdadahan-dahan. Yun yung gliding motility. Okay? So may mga mikrobyo tayong ganun ang itsura. Okay? And majority of the time that is and for the purpose of this lecture, that is Capnocytophaga for your information. Okay? Capnocytophaga. By the way, class member siya ng HASEC group. Okay? And what is the gram stain morphology of this one? It is a fusiform filamentous bacilli. Okay? Maririnig niyo yung fusiform na yan when we talk about Fusobacterium. Okay? But for gram-negative aerobic, for gram-negative facultative anaerobes, that is Capnocytophaga. Okay? HASEC, member siya ng HASEC group class. Next is another member of the HASEC group, Echinella corrodens. Okay? Echinella corrodens. From the name itself, it corrodes something. But what does it corrode? It corrodes the... ano muna tayo pala? Normal, part of the normal flora ng gingiva or bowel. Okay? So, it corrodes the agar. Okay? That's the reason why there's what we call agar pitting. So, pag narinig yung P-I-T-T-I-N-G or pitting of agar, wag na kayong mag-atubili. Okay? Ano yan? Echinella corrodens yun. Okay? Chromobacterium basipyolactium is also another one. Violasium. Okay? Chromobacterium violacum is usually found in the soil or water. Okay? And the term for the pigment produced by Chromobacterium violacum is violacin. Okay? It is violacin. Next, another member of the Hasek group, Cardiobacterium homonis. Cardiobacterium homonis is normally present in what part of the human body? It is usually present in the respiratory tract, and it is rarely isolated in cases of endocarditis, which is not rare for us, especially dito sa may pinag-workan ko. Naloka ako, meron palang na-isolate na ganyan. Akala ko dati sa libro ko lang yan nakikita. Nung nagtrabaho ako dito, my goodness. Ganun pala siya. Okay, lalo na dito sa ospital namin na to kasi ano siya, anong tawag dito? Transplant, ano siya, may transplant center dito eh. Okay? Next, we're going to talk about another organism. This is not part of the Hasek group. This is Bartonella species. There's one organism that we need to remember, but before we move on to that organism, we want to talk about the characteristics of Bartonella species. Bartonella species is a gram-negative organism, gram-negative oxidase negative organism that is fastidious, and it usually grows in blood-enriched media. Best grown in blood-enriched media. It usually goes with a cell co-culture method. Anong ibig sabihin ng cell co-culture method? Yun yung mga method, yun yung culture methods na may kasamang living cells. Right? Pero hindi pa yun gumagamit ng mga McCoy cells rin. Kaya mag-alala pang virology yun. Okay? Previously, Bartonella species were grouped as part of the Rickettsiales family or the Rickettsia family. But now it became a different microorganism altogether. Okay? And the geographic distribution of Bartonella species, or Bartonella bacilliformis, by the way, is South America. Okay? Specifically in the Andes Mountains. Okay? Next is vectors that could transmit Bartonella bacilliformis. Again, sandflies and phlebotomous flies naman lagi talaga yan. Of course, since it is a previous member of the Rickettsiales family, it could majority of the time these are organisms that require a living host to be transmitted to another organism. So tatandaan nyo lang lagi, sandflies at phlebotomous flies. And yan lang naman klase yung dapat yung tandaan na bacteria na natatransmit ng sandflies at phlebotomous flies. Gram-negative. Yeah, what I mean is it's gram-negative. Okay, next, stages of the disease caused by Bartonella bacilliformis, acute phase, or which is what we call Oroya fever, okay? And then you have what you call Verruga peruana, okay? Oroya fever is characterized by hemolytic anemia, and Verruga peruana is a chronic disease that is characterized by having a wart-like disease. Okay? This is collectively known as Carrion's disease. Okay? Carrion's disease. Doon sa mga naglalaro ng Elden Ring, Carrion yan. Hindi Carrion. Carrion. Okay? Baka maloka kayo. Okay? Next, the ideal specimen for the isolation of Bartonella bacilliformis is blood. Okay? Just like any other Rickettsiales, because it was previously known as Rickettsia bacilliformis. Okay? And then we also have another organism, Bartonella quintana. Hindi yan si Antonio Aquitania, ha? Pero ano lang yan? Ano lang siya? Other member siya. Okay? Hindi siya yung nasa bubble gum. Okay? Bartonella quintana is the causative agent of trench fever. And again, because this is from a previous member of the Rickettsiales family, it requires what? It requires a living vector again. And the vector this time is a body louse. Galis. Okay? Galis yung ano niya, body louse. Okay? Next, we move on to the next disease, which is CSD, cat scratch disease, which is transmitted by Bartonella henselae, previously known as Elizabethicium henselae. Ngayon, Bartonella species na siya. There are two causative agents of cat scratch disease. The first one is Bartonella henselae or Elizabethicum henselae. Yun yung mga previous name nila. And Bartonella clarigiae. Grabe ka naman, sir. Memorize mo pati yung previous name. Siyempre, of course, matanda na ako mentek eh. Charot. Okay? Now, IV drug users should be aware of endocarditis because of Bartonella. Okay? Ito na yon yung may previous name na Elizabethicum. Okay? Because we have Bartonella elizabethae. Okay? The reason why drug users should be aware of this one is because of the rats. Most of the time, ito yung mga na-isolate natin sa mga homeless people na IV drug users. Bartonella elizabethae. Okay? Diba? Grabe ka naman, sir. Pati ba naman yung naalala mo? Of course. Now, before we move on, we are going to give you guys a 15-minute break. This is for the anaerobic gram-negative organism. This is our second to the last discussion. I think we won't be able to finish. I will just give you guys an example. Ano tong chinat ni, whole body battery. Nakakaloka kayo. Okay, so I'll give you guys like a 10-minute break kasi syempre magtsatsaa lang din ako. Okay? Huwag mo nang alisin dyan, Patrick. Sige na nga, gawin na nga natin 15. Padadaanan lang natin lahat yan. Kasi meron pa tayong mycobacteria. Okay? May mycobacteria pa tayo. Unti lang to sana, kaso may mycobacteria pa tayo. Right? So, nandyan tayo sa gram-negative, anaerobic gram-negative bacilli. Class, wala pa tayo sa gram-positive, di ba? Grabe ka, sir. Di ba? Huwag mo na tandaan, mahaba ang spelling, hindi ko na din niya maalala. Elizabethicum. Ano siya? Ang haba niyan, friend. Elizabethicum. Kasi napalitan na siya to Bartonella elizabethae. Okay? So, chill ka lang, Lilzo. Yung ano lang tayo, yung contemporary na tayo. Okay? Hindi ko din yan nakita na tinanong sa mga exams sa mga previous kong estudyante. Mahirap ispellin. Ano ba? Okay. So, 10 minutes for everybody. Okay. Hang on a sec. And we'll get back to anaerobic gram-negative bacilli, and then we'll go with gram-positive, and then we'll go from there. Okay? 10 minutes on the clock. 15 minutes on the clock now.
Thank you, sir.
Okay. Take. Pag dyan, hindi mo naman